Good morning, ladies and gentlemen. I will be the presenter of this lecture, so welcome to the lecture from embryology and developmental biology. So on the presentation you will have the lecture, but as a material also the material for exercises that we had last year. So I didn't cut it off, so you can use the picture, for example, for your preparation. So here is recommended literature. As I said you today, lecture are pictures between 1 and 50, so I think it's obligatory, but the next pictures are from traditional practicals and they are only voluntary. Prologue. We will start with something maybe more exciting and interesting. We will speak about Nobel Prizes from this year, 2020. On the picture you can see one laureate in the left and the Swedish king. So we will speak only about Nobel Prizes from natural sciences. So Nobel Prize in physics, Nobel Prize in physiology and medicine, and especially a Nobel Prize in chemistry. You can see on the picture the Nobel Prize, the medal, and the picture, another picture from the City Hall in Stockholm. One lady is a laureate, and I think it is a lady for Nobel Prize in literature. In some years, Nobel Prize in physics is also interesting for physicians. So the result of this Nobel Prize research in physics could be applied to the medical practice. It is not the case of this year because the Nobel Prize was awarded for the research of black holes. 3 scientists gained it, so Mr. Penrose from United Kingdom, Mr. Gensel from Germany, and Mrs. Andrea Getz from USA. What is a black hole? Black hole is a region of space-time where gravity is so strong that nothing, no particle or even electromagnetic radiation such as light can escape from it. So the name is typical black holes. And here you can see the picture of laureates, and as I said you, it is not in this year applicable in medicine. Here you can see the picture of black hole. Nobel Prize in medicine and physiology is for you much more interesting. Of course, it should be applicable in medicine. So, Nobel Prize was awarded to 3 scientists, Mr. Hampton from United Kingdom, and Mr. Alter and Mr. Rice from USA, for the discovery of the virus causing hepatitis type C. Harvey discovered from the statistic of the infections during blood transfusions the existence of a still unknown type of hepatitis. So in the beginning, Mr. Harvey Alter was not scientist. He only assisted during the period of blood transfusion, but he said maybe there is one new type of hepatitis. Mr. Houghton detected the virus, and Mr. Rice studied virus reproduction and biology. On the picture you can see the particle of the virus and pictures of three laureates, Mr. Alter from the left to right, then Mr. Houghton, and then Mr. Rice. As I said this year very interesting is the Nobel Prize in Chemistry for geneticists, molecular biologists, and so on. So, this Nobel Prize is interesting from 2 points, 2 issues. So, firstly, 2 female scientists were awarded, so it is quite a rare situation. Jennifer Doudna from USA and Emmanuel Charpentier from France. So I say, vive la France. For the development of the method, it was the object of their research, development of the method of the genome editing. It's called CRISPR-Cas9. So CRISPR-Cas9 is a complex of one type of RNA that is called guide RNA, so this G is guide RNA, and the enzyme Cas9. Please, Cas9, it is not caspase, as we will speak about that in the next term. So this enzyme is endonuclease, so it means that it cleaves, it cuts molecules of DNA. So the gRNA, this is RNA part of the system, harboring the spacer sequence, helps Cas associated, so it is the origin of the abbreviation. Proteins recognize and cut DNA. As I said Cas9 is the endonuclease that cleaves double-stranded DNA near sequence PAM. So it is the sequence, it is a triplet where on the first position is could be any nucleotide, and then follow 2 nucleotides with guanine, so GG. Pictures of 2 ladies. You can see the prize is awarded in Sweden, so the first announcement is in Swedish language, and then and on the second position English. So something about CRISPR-Cas. So this system is sometimes called genetic scissors. So gRNA, this is CRISPR in this system, is the signal enabling the identification of a specific DNA sequence on the basis of complementarity. Cas9 is the endonuclease cleaving DNA in a specific site, and the genetic scissors can be programmed to find the particular site in genome and cut it out. The cut-out sequence can be replaced by another DNA sequence. The system could be used for the discharge, switching off a gene, and discover how the gene is essential for the cell or what is its function. So this second sentence, how the gene is essential for the cell or what is its function. So this approach is called knockouting of a gene. So by usage of this method, you can knock out a gene, and then on the organism you can evaluate how much is important for the organism if the activity could be replaced by activity of another gene and so on. So it is a gene knock-outing. CRISPR-Cas is an abbreviation for clustered regularly interspaced short palindromic repeats. So it is a very long name, so it's better to use this acronym CRISPR. The enzyme Cas9 is the important component of the instrument for gene editing. The enzyme is found normally in the genome of bacteria. The enzyme is an arm of bacteria for restriction, for cutting, for damage of the viral genetic information in nature. So you know that many bacteria could be attacked by different viruses, and it is an arm how to fight against these viruses in bacteria. The enzyme Cas9 in the combination with guiderin cleaves the DNA molecule in a precisely required site. Then the system CRISPR-Cas separates an undesired sequence and provides the cell with another genetic information. So by this way you can rescue or you can repair, for example, a mutation in the sequence of a DNA. Here schematically on this picture you can see the usage of genetic scissors. So a part of the DNA, a double helix, is cut off and could be replaced by another part of DNA. How works the CRISPR-Cas9? You can see this brownish cloud, so it is the enzyme Cas9. And you can see here a double-stranded DNA and guide RNA that is grayish. And on the 5' end, you can see the complementary sequence that is important as a signal for enzyme for cutting in the precise site, always near the sequence NGG that is called PAM. As each new technology or a new method, CRISPR-Cas9 has many ethical problems. After the discovery of the DNA structure in 1953, it is probably the most revolutionary discovery of molecular biology. So we can consider it as genetic correct. What are the frontiers of the CRISPR-Cas9 usage? So we can use it for the manipulation with bacterial, animal and plant DNA. Nowadays we can read some papers about manipulation with bacterial and plant DNA, but also animal DNA. For example, in plant DNA or in manipulation with plant DNA, some genetically modified organism, GMO, could be obtained, and the protocol for this technology are authorized by ethical committees. The situation in Europe, for example, for GMO is not very favorable. So in the United States, in Brazil, in Australia, it's much more easy. But what, I have the mistake there. So for human DNA manipulations, so it is much more risky and tricky, because DNA from humans, so it's something that could be from the point of view ethic criticized. So two approaches we can have. One is gene therapy. So, for example, if you use this methodology for repair of a mutation of the patient that is sick, that suffers by any genetic disease. So I think it is quite acceptable for gene therapy for cure of a patient. But of course, the method could be used for genetic enhancement of humans. So by this technology, for example, the parent, if they have enough money, can order the specific properties of their children. So, for example, desired color of eyes, height, if the boy will be tall or yeah, usually tall, not small. So we could read any papers about experiments with human embryos that were premature. So, for example, it is a case of twins, two girls in China in 2018, and it provokes harsh criticism because in this time, 2 years ago, it was very difficult to know the side effects of this technology. So then 5-year moratorium was announced for this method by application with human embryos. Of course, the experiment with bacteria, animals and plants, they continue, but there is a 5-year moratorium for experiments with manipulation with human embryos. So recently we read one article, one paper from the journal Cell, when was described another failure of CRISPR-Cas9. The paper described experiment to rescue the gene responsible for retina degeneration turned out badly. So scientists from Columbia University used CRISPR-Cas system technique in human embryos for repair the gene ICE. This gene ICE is responsible for inherited blindness, so it's important for development of retina. And they found this method inappropriate because this method caused unexpected and unwanted changes of the genetic material, often eliminating all the chromosome or its large segments. So it was published by Dieter Egli in journal Cell. So journal Cell is very famous. Many Nobelists make publication in this journal, so we can trust it. Now, something about the experiment. The experiment should prove whether the usage of the CRISPR-Cas9 is safe for gene therapy or for embryo gene therapy. Scientists fertilized egg of healthy women with sperms with mutated gene ICE, and into the fertilized egg they introduced the CRISPR-Cas9, and they hoped that the fertilized egg would use the non-mutated copy of the gene ICE delivered by the mother for the mutation repair. So the mother delivered to the zygote or to the embryo the copy without mutation, and by sperm was introduced the allele, the copy with mutated gene. So just in the protocol for this experiment was decided that the embryos would be destroyed after many analyses. So it was not expected any birth of children. And what was the results? So gene ICE repairs, the gene ICE repair succeeded in some embryos, but it failed in another embryos. The gene repair doesn't come real, and they lose large part of the genetic information. It was shown that the system CRISPR-Cas had no cut the DNA molecule in the proper site, and these lesions weren't repaired, and they started further losses of genetic information as well. So the author hoped that the results of the experiment discourage from the premature usage of the CRISPR-Cas, and they will encourage the serious research which permits its safe usage in the future. Because what is risky in this moment is the situation that we don't know the side effects of this technology as was described in this experiment. So, Nobel Prize are over, so maybe you know something new about CRISPR-Cas. And now we will continue in our lecture, and we will speak about genes in development. So the lecture is called Molecular Aspect of Development. So why we speak about molecular aspect in development? In embryology, during the studies here in Motol, in the subject embryology, you can see how the developing human being is changed in the time. Okay, you can describe some changes in differentiation, some changement of the properties of those cells. But what is important, these changes on the cellular level are provoked or are stimulated by the changes on molecular level. Especially now when we have in this position results of new methods of molecular biology, as for example DNA sequencing, Hugo project, and so on. So in the developmental biology, in molecular developmental biology, we can, for example, distinguish normal and abnormal development. So it was the reason why we speak about molecular aspect. And now, introduction to molecular regulation of gene expression and cell signalization. So two main ways in this topic of molecular development is two cascades or two directions, and it is gene expression, how gene expression is regulated, and what is the communication between cells. So embryology is a descriptive science. Developmental biology adds experimental approach and causal analysis for studies, mechanisms and causes of development of organism. So now we know that all the information for our development is saved in human genome, And very important is to know how the genes are regulated. In our genome is approximately 25,000 genes, but 4 times more of proteins. So one gene gives rise more proteins thanks to alternative splicing, and hypothesis one gene, one protein is not valid more. So in a higher organism, as for example by human, regulation of gene expression is on several levels. So maybe I'm not sure if you understand the term gene regulation. So it is the no hypothesis, but it is a reaction by which activated genes are changed to RNA, to molecule of RNA, or to molecule of protein. So this scheme, DNA, RNA, protein, could be regulated, and that is gene regulation. I'm not sure, maybe you know it from high school, but could be a little bit difficult for students in the first year as you are. So on this picture you can see some steps of the gene expression. In the upper part of the picture you can see the situation in the nucleus, in the chromosome. So you can see there the gene. Okay, so the gene with enhancer region, promoter region, and coding region. So the information in coding region is used for the sequence of the protein. The enhancer region and promoter gene have regulatory properties. So in the first step, the DNA in the chromosome is transcribed in the step 1 to one type of RNA. This type of RNA is composed from yellow parts and red parts. Red parts are coding sequences, are called exons, and yellow parts are not coding sequences and are called introns or regulatory sequences. This molecule of RNA, this is called heterogeneous nuclear RNA, is processed, so should be a little bit changed to be able to enter the cytoplasm. So the processing of the molecule of hnRNA, then the result of this reaction is messenger RNA. And then messenger RNA is transported to the cytoplasm. You can see only red molecule of messenger RNA. And there this molecule meets ribosomes, and the proteins are synthesized on these small protein factories. So it is the organelles that are responsible for the proteosynthesis. And this step, when the genetic information from RNA is translated to language of amino acid, this is called translation. And the product of this reaction, translation, is origin of a protein. The protein is not yet biologically active, but after its modification, so for example some prosthetic groups are joined something is cleaved off, a part of the sequence is cleaved off, so the protein could be methylated, glycolated, and many other. And in the end, you can obtain a protein that is biologically active, and it is a main product of a gene. It is a gene expression, how the gene is expressed, okay? But sometimes the gene expression can terminate by the formation or production of RNA only, yeah? So please don't forget it. It is also important that the product of a gene is not only protein, but also molecules of RNA. What is typical for development, it is differential gene expression. So this gene expression, differential gene expression, it is a gene expression in time and space. So never total gene expression in one moment and on one place. So during the development, only some genes are switched on, and only in sometimes very short time periods. We can also speak about hierarchy of developmental genes. So some genes regulate the activity of another genes. For example, there is one example of two groups, very important groups of developmental genes, maternal genes and zygotic genes. The names come from the place of expression. So maternal genes, they are expressed just during maturation of oocyte. And for example, some typical are bicoid, torso, caudal, hunchback, nanos. And the second important group are zygotic genes. There is also hierarchy in zygotic genes. The zygotic genes are expressed just in zygote or in very early embryo. So it exists also hierarchy in zygotic genes. For example, the first activated are gap genes and PAX genes, segment polarity genes, and Hox genes. So these four groups of genes are called zygotic genes. They are expressed in the zygote. What processes on molecular level we can meet during this process. So maybe you know from the enzymology that there is a hypothesis of log and K. In active site, the molecule of substrate fit as K to a log. So also between enzyme and substrate in enzymology we can find it, but also the interaction between protein and nucleic acid, for example transcription factors and DNA. So these two molecules, protein of a transcription factor and the nucleic acid, as for example DNA, they can interact, they can be bound by not very strong bonds. But of course two proteins could interact. So for example a ligand, if the signaling molecule is a protein or peptide, can communicate with its receptor, that is also protein. But we can find another interaction between nucleic acid and nucleic acid, so DNA, RNA and DNA, RNA and RNA. So these interactions are called hybridization of nucleic acids. In this case, we have to consider three-dimensional complementarity of the molecules. So that is a hypothesis of log and K. Here you can see it. For example, the K is substrate, log is enzyme, so they correct fit and will react. If they don't fit, there is no reaction, the enzyme doesn't work. Very similar, for example, on the level of another protein, not only substrate and enzyme, but also two proteins, two molecules of nucleic acids. This signaling complex we can see also in this picture. Sorry. So here you can see on the cytoplasmic membrane a receptor and signaling molecule that is bound, so a receptor is activated. And then you can see the transport of the signal downstream in direction to nucleus. So if these molecules fit as K and log, as we can see here, the signal could be transported or transferred very easily. So this blue molecule is called scaffold protein. So if in absence of this protein, there won't be so fast communication with the signal. Very similar it is here on different receptor as it is here. So this interaction between two molecules in regulation of gene expression or in signaling are possible only if the molecules can fit, as for example these molecules fit to scaffold protein. Why these molecules have the affinity each other? So again you can see here the receptor and some signaling molecules. Usually, we will speak later about that, it can be the kinase activity on the intracellular part of the receptor. So kinase activity, it is enzymatic activity that can phosphorylate another molecule. So add the phosphate group to another protein. So Y is tyrosine, and on tyrosine, because it is amino acid with hydroxyl, could be added phosphate, and this phosphate can interact with domain that is called SH2 with on another protein, okay? And this greenish protein have another domains, for example plextrin homology domain or phosphotyrosine binding domain as here. So, and it can react with another proteins, as we can see here. For example, adaptor protein has no enzymatic activity, but has two domains, SH2 and SH3, and can connect another signaling protein, as we can see here. Why this situation is for students quite difficult, but why we speak about these interactions? because sometimes, for example, in medicine, it is necessary to block these signaling pathways. As if you know some inhibitor of this interaction, you can stop the signaling cascade. So what is interesting on development, that only several basic principles to start and to make sense of developmental process. So these developmental processes are evolutionary conserved. Okay, so if we can study it on Drosophila, for example, we can find similar proteins in human being, or in human genome, for example. And the interaction that we describe can determine how the embryo develops. We will speak about gene regulation, how is influenced expression of a gene, but the second very important process during development, it is a communication between cells, cell communication, cell signaling, okay, and in embryology it has special Nobel name, embryonic induction. So it means embryonic induction, it is normal cell communication or normal cell signalization when one cluster of cell can make induction of a property in the another group of cells. So you can see here embryonic induction. The influence of one cell group that is called inducers over another of neighboring cells group. This group are cells, induced cells or competent cells or target cells. And this interaction is very important during development. The competent cells, the acceptor of the signal, should have receptors, okay, and signaling cascade. We will speak about these molecules later, and you can find also some information in our e-learning program. So the competent cell should obtain two types of molecules, receptors and signaling protein for signaling cascades. There are two types of embryonic induction: permissive induction, where we can expect only one kind of response to a signal, and if a given signal level is not reached, there is no signalization. If it is reached, the signalization can occur, so we speak about the threshold effect. The second is instructive induction. The cells respond differently to different concentration of the signaling molecule. This signaling molecule that have this property to have different response on the basis of different concentration are called morphogene. They are not the gene, they are only signaling molecules, if the name is a little bit similar. So what are the phases of the intercellular signalization? So what will be the result of this signaling processes? So after the response of the signal, for example, it could be changed contractile physical forces between cells, position in germ layer. Very important is position information, and for example, it could stimulate activities of adhesive molecule. It can trigger apoptosis and so on. So there is, I said you that the information for development is saved in DNA, but also epigenetic effect is important. Epigenetic effect is based on the modification of the bases in the DNA. So a picture for girls, you can see two boys, they are the twins, they are the monozygotic twins, as you can see. They are very similar, but still they are different. So the boy in the left, so it is a sportsman, passed more time doing sport. The second one passed majority of time in libraries in the university, so he is a little bit more tiny. So, and how this, because genome is identical by both, but why they are a little bit different. So it can be caused, for example, by nutrition, it can be caused by sports, and so on. So these types of changement of the phenotype are epigenetic. Usually, these epigenetic changes are caused by methylation of the DNA. Only one nucleotide could be methylated, so methyl group could be added to this molecule, and that is it is cytidine, so with the base cytosine. Okay, only this base could be methylated, but it changed slightly the genetic property, and sometimes is considered as fifth base in the sequence of the DNA. So please, not only genetic, what is coded in our genome, but also influence of environment that can change development. Back to the regulation of gene expression. I said you they could occur in different levels. So, for example, in the beginning, different genes could be transcribed. So, on the level of transcription. So, for example, for this transcription are important protein molecules called transcription factors. And of course, if there is no transcription factor, no transcription occurs. So, primary transcripts could be selectively processed. So, for example, you can obtain from one gene different types of messenger RNA. Okay, so it is called RNA processing. So it means, for example, capping, polyA-tailing, and also the splicing of this molecule. Very important role play here the specific types of RNA that are called non-coding RNA or interfering RNA, that is then expressed as RNAi. These RNAi are very small molecules of RNA. They code for nothing, but they are complementary to some parts of messenger RNA, and by this way they could destroy. So if you have no messenger RNA, no translation. So it is also one level of regulation. Very similar could be also selectively translated different molecules of RNA. Again, one type of RNAi could regulate this process. And when the protein, protein is formed on the ribosome, this protein is not in the beginning biologically active, and they could be processed specifically as well, and you can obtain, for example, protein with different glycosylation, with different content of ions and so on. So on all these levels, the development could be changed, the gene expression could be regulated. It is only repetition here, so something about gene transcription. You know that genetic information is saved in chromatin. The DNA is bounded by nucleosomes that is composed from histones, proteins, and there are two types of chromatin: heterochromatin that is non-active, and there is no active transcription, not very active transcription. Euchromatin, it is an active part of the chromatin, and it is active form capable of transcription. What is the gene structure? I think we will have the picture. Okay, so gene obtains promoter region. There are some very short sequences, for example, Tata boys, Cat boys, CPG islands, and this part of a gene, it is a regulatory part of a gene, is important for interaction with transcription factors. Transcription factors are always proteins, so that have two very important domains: DNA binding domain and transactivation domain. Another part of a genes are enhancer, silencer, and so on. So on this picture you can see structure of a gene. You can see yellow part is a promoter, for example, with the specific sequence Tata box. Then there are the green parts, they are coding sequences. White parts of the gene, they are introns, they are not spliced, okay? And you can see, for example, in intron 2 is inserted enhancer. It's a rare situation, but could be. And there is also another signal, for example, for initiation translation, termination translation, poly-A tailing, and so on. So that is the typical structure of a gene. During discussion about regulation of gene expression, we will speak mainly about promoter. You can see promoter you can find very often on the 5' end of a gene. But there are also another ways of gene expression regulation. I said you, for example, splicing. You can obtain different isoforms of the spliced protein. Example is gene or protein WT1. It is a gene important for development of kidney during embryonal period, and for example it is switched on during organogenesis only 2 weeks, and then is switched off all over the life of the organism. But in pathological conditions, of course, they can be switched off, but then, for example, some type of cancer could occur. So it is an alternative splicing that can also express, regulate regulate expression. I mentioned non-coding RNA, small molecules of RNA that don't code for anything. For example, they are called, for example, microRNA, small interfering RNA, or RNAi interfering RNA. Another side of the gene regulation and development regulation could be post-translational modification, as for example cleavage, phosphorylation, combination with other proteins. So some peptides are subunits of a larger complex. Of course, protein transport is also important. Glycosylation, ion equipment, prosthetic group equipment, and so on. So in the development you can find many regulation levels. On this picture you can study alternative splicing, or you can understand alternative splicing. So normally in the gene there are 4 exons, 1, 2, 3, 4. And in the transcript, in messenger RNA, you can find also 4 sequences complementary to exon 1, 2, 3, 4. But it can occur alternative splicing, and in the first protein are spliced only first 3 exons, and in the second part exon 3 is missing. The protein, biochemically, they are very similar, but they are not identical. So we obtain protein A, protein B. Very often these proteins form protein families. So it is a set of the similar proteins. Not always have the same biological activity, not always have the same biochemical activities, but they form protein family. Another side of the regulation, for example, on the level of the chromosomes. So you know what is the nucleosomes, what are the histones, and also by this way, by function of acetylases and deacetylases, we can regulate gene expression. To perform transcription, the DNA should be unloaded. Okay, so if histones are acetylated, you can see here the acetyl group, then the original positive charge of histones can be neutralized. They could change the charge, could be neutral, for example, and then DNA is not bound with these histones in nucleosomes. This process is irreversible, so as exist the enzymes that acetylate and permit the unloading of the DNA, exist also the acetylases that cleave off this group SH3CO, and then there's a formation of the structures that are usual from chromosomes. So it is one level, the level of the chromosomes. But there could be some modification also on the level of the promoter, and it is gene methylation. And we mentioned some specific short sequences in promoter, for example, as CPG islands. CpG islands are dinucleotide with bases cytosine and guanine. It's small p. It is sugar phosphate group or part of the molecule. Okay, what is a little bit special by CpG islands? These CpG islands are accumulated in the sequences of promoters. So in the beginning, in the period of sequencing of a gene, for example, we obtain the sequence of our gene, and it was interesting to know if the gene is coding genes, if codes for a protein. So if in this sequence was found accumulation of CpG islands, so the sequence CG, okay, so it was very probable that the sequence is coding for some protein. So this makeup of C is made by the activity of methyl transferases and also demethylases that cleaved off the group CH3. And all these two processes are important in, for example, cellular signalization and regulation of gene expression. If we speak about CpG islands and the regulation of the gene expression, we can mention two groups of genes, so-called housekeeping genes, for example, genes coding for tRNA, ribosomal RNA, heat shock proteins, beta-actin, and so on. And luxury genes, for example, WT1 that I mentioned. So that genes, this luxury gene, are important for development. So what is the function of the housekeeping gene? Housekeeping gene maintain everyday activity of the cells, for example, metabolism, replacing some parts of the body of the cells. Luxury genes are switched on only if there are some, for example, changement and of differentiation and so on. And for example, by models, genetic models, as it is Drosophila or Caenorhabditis elegans, this developmental genes, these luxury genes, form 10% of all genes. This luxury gene codes for receptors, the ligands, growth factors, they are signaling molecules, GF, growth factors, intracellular signal molecules, protein regulating gene, and many of them are expressed only in embryonal period, for example, SWT1, as I mentioned it. What we can tell about the promoters of housekeeping gene and luxury genes? So, housekeeping genes, they work permanently. Maybe not on very high level, but they work permanently. So it means that their CpG islands are not methylated because the transcription factor are joined to this sequence and stimulate transcription of the gene. On the other side, luxury gene that are, for example, active only very short time during the development and the life of the organism, they have CpG islands methylated. Yeah, I spoke about this. One note to regulative and mosaic embryos or regulative eggs and mosaic eggs. During the development, we distinguish two types of very early embryos. So, one type of embryo is called regulative, and they are very early embryos, and in these embryos all cells are equal. So, for example, if we take off one or two cells of this embryo, very early embryo, nothing happens because the embryo can replace these two cells that were, for example, cut off from the embryo, these two missing cells. So in this time, in the time of regulative embryo, all the cells are on the same level and they are not specialized. Okay, so they can give rise to all cell types in the human body. So it is considered that is approximately 200 cell types in the human body. We call them, the cells we call totipotent, and for example they could be used for cloning of an organism or for pre-implantation diagnostics. On the other side, the mosaic embryos, they can develop only according to their early fate. So later during the development, the embryos are sets of two type cells, totipotent cells, and also the cells that were partly differentiated and specialized. So demarcation between them is not very clear, and but this changement from mosaic to from regulative to mosaic occurs very early. So we can also manipulate with embryo, but no for cloning and pre-implantation diagnostics. How was discovered cell communication? So you can see here, you can see here a picture of a very, from the point of view today's level, very primitive experiments that were performed maybe 100 years ago, when this very primitive instrument, that is a scalpel, was transferred different cells of embryo from one part of the embryo to another, and was studied their development. So you can see, for example, here embryo with two regions, region A and region B. It is as a fate map, and just embryo differentiated. So the yellowish cells give rise to hexagonal cells, and the greenish cells give rise to cubic cells. If the cells from the domain B are not determined, and you can transplant it, for example, then if they are not determined, they develop to hexagonal cells, as it is in region B not determined. If the cells in the region B are just determined, specialized, differential, so if you do the transplantation among yellowish cells, the cells will develop as a cubic cells. Okay, so it is the region B determined. And there is a control expression region where is specialized, and in the culture it will develop to cubic cells. So how it is possible that we can arrange this experiment? So it is caused by chemical signals, signaling molecule, and as I said you in the beginning, it is embryonic induction. Types of cell signaling. Three main types of signaling. So, signaling by diffusion. You can see one cell is producing this signaling molecules, the orange one, that are accepted by another group of the cells through the activity of the receptor. And then in the cells are formed second messenger. The biochemical character of a signaling molecule that is produced by one cell type to signaling molecule in another group, I call it second messenger, is different. So the cells are different. Usually the first type, the primary messengers are, for example, peptides or proteins, and the second messenger are very small molecule, for example, CAMP, cyclic AMP, for example, phosphate, calcium, and other molecules. So it is signalization mediated by diffusion of signaling cell. Another type, for example, it is signaling by signaling molecule. Signaling molecule is red on this picture, but is fixed on the surface of the first cell. So if the cells are neighboring, so the ligand, red, and the receptor, blue, can meet, and then again in the cell is stimulated production of a new signal. It is the same type of the signaling molecule. It is second messenger. And the third type of the signalization, it is signalization mediated by gap junctions that you know from embryology. Very simple scheme of embryonic induction. So secreting cells produce the red molecules that are chemical signals, and these chemical signals are accepted by the receptors of target cells and can change the gene expression, the pattern of a gene expression. So in the left part it is a signalization, for example, synthesis and release of signaling molecule, and in the right part of the picture it is a reception of signaling molecule. So it is really very simple scheme of signalization. On this picture maybe it is in more details. So you can see the signaling molecule. It is sometimes called ligand, sometimes it is a primary messenger. Usually primary messenger are bigger molecules, as for example peptides and proteins. They cannot enter the cell, so they communicate with the cell via receptor. You can see that the receptor and binding of the ligand by this receptor. In the intracellular part of the receptor, you can see the reaction where second messenger is formed. And this second messenger can influence another processes in the cells and can, for example, trigger the signaling cascade directing to the nucleus and there change the pattern of gene expression. I think it is the most complex picture of this lecture. So we can hear different type of receptors in cell signalization or in embryonal induction. So the picture A. Here it is a signalization, for example, with steroids, steroids derivative of retinoic acids. So these molecules, signaling molecules are relatively small and easily can enter the cell. In the cell, as here for example, in the cell they meet receptor. In this case, HOP protein 90 is fixed to this receptor, and the ligand can bind to this complex and can change conformation. And in this new conformation can enter the nucleus and regulate gene expression. Okay, so it is a very rare example in the nature when a receptor that binds ligand can be also in the same cell transcription factor, so can influence gene expression. So it is typical only for small lipophilic molecules as, for example, steroids. And in the next pictures we can see the receptor that is bind to cell membrane. So picture B, we have there the enzyme link receptor. The ligand cannot enter the cells but can be bound to receptor. And for example, on this intracellular part of the receptor, you can find kinase. And this kinase, it is the enzyme that phosphorylates. Kinases are enzymes that add to the molecule a phosphatic group. And by this linkage between ligand and receptors, these kinases could be activated and can phosphorylate another molecules in proteins, I mean in the cytoplasm. We have two types, or we distinguish two types of kinases. They are the tyrosine kinases, and the second group is serine-threonine kinases. Tyrosine kinases, tyrosine, serine, and threonine, because they are the amino acids that obtain OH group. For this OH group could be add phosphatic group. So it is an enzyme linked. It is a receptor with linked enzyme. Picture C is not very popular by students, but the situation is similar. Ligand cannot enter the cell, could mediate the development by activity of the receptor, and on this receptor are bound from the inner part, can bind G proteins. It is here on the picture GI these G proteins, why they are called G proteins? Because on their surface is bound GTP or GDP. I hope you can imagine ATP. Very similar to ATP is GTP. Okay, so this G protein can bound on the surface GTP or GDP. If GDP is bound, nothing happens. But if GTP is bound to this surface, then the G protein dissociates to some subunits, and one of the subunits can activate adenylate cyclase. Adenylate cyclase is another enzyme that can produce from ATP cyclic AMP. And you know that cyclic AMP, it is very potent second messenger. It can have different effects in the cell for signalization. Next, continuation of this picture. It is again receptor linked with one type of G protein. Here it is GQ. Again, ligand is bound to the receptor, and GQ, this G protein, is activated, and again one of the subunits can activate another enzyme that is called phospholipase. Lipases are enzymes that cleave lipidic molecules. Okay, for example, as it is phosphatidylinositol bisphosphate, PIP2. Okay, and this phospholipase C cleave the molecules of phosphatidylinositol bisphosphate to diacylglycerol, that is alone signaling molecule, and inositol triphosphate, that is also signaling molecule. Especially it activates in endoplasmatic reticulum the calcium ions. Okay, so then, for example, the effect of signalization is multiplied. So this is two types of the receptors linked with G proteins. And the last picture is quite easy. Here we can see a receptor that is linked with ion channel. If, for example, ligand is bound to the receptor here, then the channel is open, and, for example, calcium ion can enter the cell. So here we have discussed different types of cell signalization, and in this cases B, C and D, we mentioned some different types of transduction of the signal. So ligand approaching to the cell cannot enter the cell, so it is a primary messenger, but it can stimulate the production of the second messenger, for example, a cyclic AMP. So the quality of this, for example, growth factor, primary messenger, is different with this cDNA molecule. I think now that's all from our lecture. So here on this picture you can see one of genetic models. It's called Caenorhabditis elegans, and it is soil worm, not longer than 1 millimeter, and it's used for many experiments in genetics, in genetic experiments. Okay, so thank you for your attention. I wish you Merry Christmas, because I think we won't meet again, and I wish you many successes in your life, in professional life, and in your personal life. So, in the next pictures, you can use this picture for studying something, but these pictures are only voluntary, are not obliged, but you can find some information there, and it was the original, it is a original presentation that was discussed in practical exercises, in practicals. But this year it is a little bit different. So please, for the preparation for your test, use the lecture and also the e-learning program that is on Moodle. Okay, so again, thank you for your attention and good luck.